Namaskar. Welcome to Prakash on Basics. I'm Prakash Zog. Our most important sense organ is obviously the pair of eyes that we have. Try to carry out your normal activities for about an hour without opening them at all, and then you'll realize their importance. Naturally, understanding them is very important for all of us as human beings and as a part of things that we must know all around us. We break down this information in two or three steps because it's a very vital area. I'll first explain its structure and the basic working. Realize that our eyes form a specialized sense organ, obviously a pair. What it does is it creates images using light energy, which are interpreted by our brain, and then we get an understanding of what things are around. It's more like an optical device, more like a handycam that you have nowadays, or previously as it is to be called as a simple camera. It's not really all that simple. All human beings, basically vertebrates, have a pair of eyes protected from mechanical injury by a set of seven bones that form a socket on either sides. Um, they're on the same side of the face. You don't have one behind, one in front. So a pair of eyes separated by centers by roughly around 3 to 4 inches, and that's roughly the distance between the two of them. And they are all controlled by motor nerves for all the possible movements that are required. Realize that eye is among the earliest organs developed in the fetus. Um, and when the child is born, it's not fully developed. Originally, at the final stage, its size is. Let me give you the mechanical dimensions. It's a sphere that is slightly squashed on top, elongated to some extent, but roughly its diameter is around 0.93 inches, about 2.3, 2.4 centimeters in diameter. And its volume is roughly around 6 cubic centimeters. The size might vary a little bit because at the birth, these eyes, which are a fairly good set of organs, is around in terms of diameter, it is around 16 mm. By the time you are around 3 years of age, it has grown to around 22 millimeters. And by the age of around 12, it has reached its maximum capacity, which could be actually a little more than 23 and close to around 25 millimeters. That's 2.5 centimeters. Each is of a size slightly smaller than a table tennis ball, but it's more like a tennis ball in the form of layers. It has multiple layers, minimum three that we have to differentiate and know about. The outermost one is known as the sclera. It is whitish. The next one is known as the choroid, and the third inner one is the cornea. Uh, that is, it is added with some special layers at different places, and these have an innermost important layer, which is the one that many of us know of, which is the retina. The sclera is a tough protective outer shell of the eyeball, each of them, obviously. It's dense, fibrous tissue. Um and it is the one that provides all the attachments inside and to the sides also, with a number of muscles. They are more like a set of fingers that are attached from all sides. Roughly around seven of them are there, and these are the ones that control all the possible movement of the eye that we know of. This sclera itself from inside extends and comes to the part that we see, which is the surface which is known as conjunctiva. And this is that transparent part that is a protective layer on top of our front part of the sclera. So it's more like a ball. One sixth of it is visible from the front, and it is held by a set of muscles from behind, like like a cricketer who uses or manipulates things by using his fingers. The conjunctiva is transparent mucous membrane, and that's the one that prevents our eyes from getting dry. Um, Inside, now the outermost shell was whitish sclera. Inside it, there is a thick, light-proof layer, and that is the choroid. Realize that the extension of all these, the sclera, results into a very important term that most of us know of, which is the cornea. It is dome shaped, about half an inch in diameter. It's transparent, and it's in the front part of the eye, that is visible to us. It's a very sensitive surface, very sensitive to pain, very sensitive to touch also. Um, and it's curved. It's more like a biconvex type of a system. Um, it has only blood vessels in the peripheral region, that means in the outer region, and the inner region doesn't have any blood vessels. So all its nourishment, oxygen, etc., is provided through the tear glands and the tears that, or the wetness that we have in our eyes. That's the one that is the fluid itself is antibacterial, and therefore the eye is free of all sorts of germs or other factors that can affect the eye. This cornea has two major functions. The first, it protects the next two parts which we know of, which is the pupil and the iris. And more importantly, it is the first step in refraction that takes place, which produces the final image inside our eye. So it's more like something like this. That's the type of surface that is there. And it's transparent, and it is on top of our eyes here. Um, the cornea is also a barrier for infection. Uh, if it becomes opaque, or if it gets damaged or clouded, um, there could be a lot of problems, and what they are is something that we will talk of next time. Then that colored part that you see inside is typically referred to as the iris. And this iris has a very small opening, and that is the one that is known as the pupil. This iris and its colors are associated with our genetic code. So each of us have a typical color to our eye, depending upon our genetic makeup to a large extent. We must also realize that the basic function of this iris is to control the opening that is there in front. So there is a set of circular muscles, sphincter muscles as they are known. There are some special places in our body where we don't have normal muscles like this, but we have muscles that can contract and make openings. Our throat, our gullet, and various other places where we have such muscles. One of the most important ones is there, and that muscle is the one that controls and reduces the size of the opening or increases. And that's when we say that it dilates or is constricted. How and why is something that we must know of, and I'll come to that. Now, there is one more aspect. Cornea, iris, and all that area. is followed by a crystalline transparent lens. It's a bifocal lens. We talked of lenses, so you are aware of what a lens is. Bifocal means both sides are curved and both sides are outward, convex, biconvex lens. And this biconvex lens is pretty fairly small in size. And it is what's very important is that it is flexible. So its shape can be modified to a small extent. And that modification of the shape is going to cause a change in its focal length. To achieve that, on either sides, above and below, we have a set of muscles which are known as the ciliary muscles, which can actually stretch and relax a little bit. And when that happens, the eye lens can change its shape to a very small extent. So, the lens, the parts in front, and in between them, there is a small region that is known as the anterior chamber in which there is a fluid. And beyond the lens, within the eyeball, there is another jelly like substance. And both these are equally important. One is known as the outer one, is the watery type of liquid that is there, which is known as the aqueous humor. Names, you must have just heard about them, that's all. And the inner one is known as the vitreous humor. Vitreous means a little more dense, so it's more like a jelly, and that is the one that ensures that the shape remains perfectly round and so forth. It contains water and proteins and so forth that are required for internal nourishment, etc. So a sphere, a lens on one side, and this inner region here is the one that has a special area made of nervous tissue. And this layer is the one that is known as the retina. It covers practically two-thirds of the back part of the complete eyeball inside. And it is connected to the brain by a very important nerve, which is known as the optic nerve. The communication is very important because a lot of information goes across. And from inside, it is so close to the brain that many a times it is the retina is referred to as an extension of the brain, because that's a very important sensing part and information collecting part that we have. This retina itself is multi-layered, uh, and only the outermost one is the one that is photosensitive. Photosensitive, photon is light, and sensitive means it can react to that. It's basically a chemical change that might take place, and that is the one that is triggered, and that is the one that sends some signals. Neurons, remember what I talked of long back. So this is also a neuron. So, and these are typically then known as photoreceptors because they receive light energy, convert it into nerve signals, and send them to the brain. But then there are two types of nerve cells that are there. One is known as the rods, and the second one is known as the cones. That's because of their shape. Rods are elongated and long, and the cones are conical in shape. Uh, how many? It is estimated that there are roughly 130 million rods, and around a little less than that, which is around 7 million only cones. The cones are concentrated in the central region of this retina. There's a small depression inside, even in that curved part, which is known as foeva. And the concentration of these is maximum there because if you look at the axis, if you go inside, the point where the rays, a straight set of rays of light would hit is the region where the concentration of these cones is maximum. And around it and spread out in general everywhere is the rods. So you have the rods and the cones. And what do? The rods are the ones that are sensitive to the intensity of light. how bright the light is or how weak the light is. They are specialized neurons, and they are the ones that help us to decide on the shape, size, brightness of the image. But they are more related to black and white images because they are associated with the intensity of light. And what causes all these changes is something that we know of. The complicated name for it is Rudolph spin, but we know it as vitamin A, and that's the one that is essential to bring about the necessary changes that send the necessary signals to our brain. And the cones are the ones that are associated with the color vision that we have. They are less sensitive to low intensity. That is why at night, if the light is less, you can't see colors. You can see what you can see is because of the rods. You can see, but then it is more of a grayish shade, dark gray or light gray. That's about it. You can't really see colors when the intensity of light is very low. As I said, they are concentrated more in the central region of our retina. The cones themselves are split up. They are divided into three different types because they have three different sensing pigments inside. And these pigments are associated with different colors. Um, we talked of light. We are aware that it is the wavelength that decides everything. So there are certain cones that are sensitive to long wavelengths. And the long wavelengths are red light area that we have. Then we have the middle wavelengths, which is associated with the green light. And that is the one that is absorbed by these and sensed. And the short wavelengths, which is blue and violet. And when these are absorbed, they produce the necessary signal in these particular cones. So red, green, and blue. Is that RGB? And have you heard this word somewhere? I'm going to talk of it sometime later. Realize one more thing. When I say it is sensitive to a particular wavelength, it's not just one wavelength. That's the peak area, and that is a spread. So, in fact, there is one peak, second peak, and a third peak to which these cones respond. But the light that comes to us is of different colors. And the different colors, let me remind you, is because of the percentage mixture of these different wavelengths. And so the amount of intensity is also recorded to an extent, and the color is also recorded. And so the combination of the two gives rise to the typical color that our brain can then sense. And this then has to be carried to the brain. That is done by the optic nerve. And the optic nerve carries this information about the image in terms of its intensity, in terms of its color contrast, and therefore naturally whether it is a bright or a dark image or a black and a white image and so forth. It carries this information to the visual cortex of the brain, and that's the area in which vision and our image formation and detection of what we see is interpreted. The optic nerve has to go from the retina to the brain. The point from the retina where it enters and goes inside is an area which would have no sensing units. It wouldn't have rods and cones. So naturally, if the image is formed at that point, it cannot be seen. And that is typically referred to as the blind spot. So the blind spot is the area on the retina on which if the image is formed, it cannot be sensed because that's the point of entry of the optic nerve. So what happens? This is what the structural pattern is. So light falls on an object. It's transmitted back towards you. It comes along the axis, let us say. First, it falls on the cornea. There the first part of the lens job is done. So part roughly around 40% of the bending of light that is required is carried out there, and then it falls on the iris. It has to go inside through that small opening that is there. But if it's too bright, the iris constricts and limits the amount of light that goes in. If it's a bit dull, it opens out a little bigger and lets more light go in. And so that size opening and closing, that is typically referred to as the aperture. The aperture increases or decreases and the iris goes in, and the pupil is the opening, and the iris is the diaphragm, that or the arrangement that changes the shape, and the light then falls on the crystalline lens that is inside. This crystalline lens, uh, how powerful it is. Realize that the eyes, I told you, it's pretty small. So the lens must be pretty well shaped and it should create that necessary image. Its number, diopter value, is close to about plus 18, and that's the one that produces the image. What is diopter value? Yes, I will tell you about it because that's associated with our spectacles that we tend to use. I'll come to that. But the image then is formed because of this combination on the retina. The retina then senses it and carries this information through the optic nerve towards the brain. Realize that because it's a lens, it produces an inverted image. Because the image is real, it is um, diminished because of the distances. And then obviously I can't see an inverted image, so the brain has to reverse it around. Rather than that, the optic nerve itself splits the information into two sets and then passes it on to the brain. And in a particular region within the on the way to the brain, the image is inverted. It is straightened out and sent to the brain. Realize that if you close one eye and then the other one, there is a slight difference in the images that you would see. So naturally, the information is split accordingly and sent to the brain. It has a special purpose of its own. And then that image there is interpreted. And once the image is interpreted, you see the object that we are talking of. Depending upon the amount of light that is coming, the pupil will dilate or uh, uh, constrict itself. Uh, the image originally is 2D, but we seem to see things in 3 dimensions and interpret things. How that is done is one step ahead. The area in which this crossover takes place is known as the chiasm, and the processing actually, as I told you, takes place in the visual cortex. The crossover is the one that results into the binocular vision that we know of. And this is how the image formation takes place. Realize that very small children can't really see things very clearly because the eyes have not been the muscles have not been coordinated properly. The image formation is not precise, and so tracking is not very good. But by the time we grow up, our muscles have learned to track things properly. So the pair of eyes will track the object wherever it is and ensure that the image falls exactly on that high concentration level point forever, where the image formed is the best, maximum clarity. So realize that when you're looking at an object moving, and if you want to look at it, you turn your head. And if you can't turn your head, you at least turn your eyes. The reason is you turn them in such a manner that you ensure that the image falls on that central, more concentrated area where the image is the best. So when you're reading or doing very checking up diamonds and things like that, that is the time when the image has to fall precisely on that particular area. But the rods and cones have the rods spread all around, and that outer region is the one that senses in the peripheral region of our vision. What the range is, how the image is formed, how it is modified, what are the different problems associated with it, all that is equally important, because our eyes are our most important tool for getting information. That's a lot of more information, and we need to discuss that also. If you have liked whatever I have said, please like and subscribe to this channel. Tell your friends about it. Look at some school diagrams that you must have done sometime about the size of the. It's very difficult to realize it's just a small ball, one here and one here, that is giving us so much of information. And the beauty of how it works is something that we need to discuss next time. We also need to know what their problems are, because we need to take care of our eyes. Till we do that again next to next week. Namaskar.